Okay, can someone confirm that you can hear me?
Yes.
Great.
Yes.
All right, today is January 17th, and we're supposed to be finishing chapter 2 and starting chapter 3. Let me inform you that you have your first real quiz. So make sure you have Respondus Lockdown Browser up and running on your computer and use it to take the multiple choice quiz, which is also named Quiz 1B. You need Respondus Lockdown Browser in order to take that quiz. The quiz must be taken sometime between, I think it's 8 p.m. Tuesday night to 8 p.m. Wednesday night. Let me double check on that to make sure I got that right. This will be the first quiz, so make sure you have Respondus Lockdown Browser working on your system before attempting to take it. There is also a practice quiz 00A. It is a fill in the blank answer or fill in a short answer. And that one does not require Respondus Lockdown Browser to take. It is not a timed quiz. You may take that one anytime between 8 p.m. Tuesday to 8 p.m. Wednesday. And you can take the entire 24 hours. Making sure I got that right. But for the multiple choice quiz, meaning quiz 00. Wait a minute, am I on the wrong? I might be on the wrong thing. What am I talking about here? There's one. I'm on quiz 00. Let me close that. Once you begin this quiz, you will have 25 minutes to finish it. That's 1 minute for each question, and then something like 3 to 5 minutes extra for the entire quiz. So once you begin between 8 p.m. Tuesday night to 8 p.m. Wednesday night, you have to finish it within 25 minutes. Any questions about the quizzes?
I missed what you said about the attempt. Was it one attempt for the lockdown quiz?
For all the quizzes except for the practice quizzes, you only get one attempt on it.
Okay.
Okay. For the practice quizzes, you did get 2 attempts on them.
Okay, do we still get credit for the practice, right? For the practice quiz?
You will get credit for the practice quiz. I've got to go in and manually give you the credit, meaning it doesn't send it to you automatically for the practice quiz. I should look into doing that because I do have it set up where it could do it itself. But I'll go in and manually give you the credit.
Okay, thank you.
I haven't graded the quizzes yet, so I don't know.
I don't remember if it was this class or another class, but I just want to ask anyway. Does the lowest quiz get dropped or no?
The lowest quiz does get dropped.
Okay.
Not counting the practice quizzes.
Okay, thank you.
All right. All right. About 2 days after the quiz shuts down, you can see the answers for the quiz 1B or all the quizzes B. You will have to go into that quiz with Respondus Lockdown Browser to see the answers. For quiz 1A, you can come to this link. You can't see it now, but that should be available starting January 21st. And then the answers will display for about 1 week, after which time the answers will become locked again. You can't get to them. All right, is there anything else I need to talk about? Let me remind you, you can form an extra credit study group. You can get together and do it in person. That's the way students always did it before the pandemic. On the online classes, most students get together with Zoom or some other software and then meet. You do need to study for at least a half an hour. I prefer you to quiz each other, but that is not required. And just click on the link and it'll tell you what to do to get the full extra credit point. You do need to tell me everybody in your group, the full names. Tell me when you studied, the date, and what you studied, and for how long you studied. When you're talking about what you studied, you just need to briefly talk about it. You don't need to tell me in great detail what you studied. You may do this for each week for a total of 9 points, 1 per week. You may not do your lab. You are supposed to do the lab during your lab time. So you're not going to get credit for both a lab worksheet and then studying for the lab. The extra credit will be out of 0 points, and then it'll be 1 point out of 0. So that's 1 point of extra credit. You do need to study for at least a half an hour and then submit it to this, what do you call it? File or page here. And you'll have a link either on the right here or down below to upload it. I've got a speed grader link. For this week, we do have some optional metric quizzes. If you want to test yourself on the metric system, you will be quizzed on the metric system, but you do not have to take the metric quizzes. They're just for your own amusement or your own enjoyment. And if you want to practice the metric system, let me warn you that you can copy the link there, or you can click on it. And if you click on it, it won't open directly in Canvas. So you got to click on it a second time, meaning there will be a link up there. If you click on it here, it'll then open it outside of Canvas. And let me see if I can remember how you do this. You have to be careful what links you push on this page so you don't download something. I think we actually push start on this one. And then there's the first question. Select the base unit of the metric system for length. And that one I know is meter. If it's green, it's correct. Let's go on to the next. Don't do the download, just click the next. There it is. And we're going to select a wrong answer here. Select the base unit for the metric system for volume. And we're going to say, there it is, kilometer, which is incorrect, and it'll show red. It'll also tell you the number of questions you answered and the percentage of correct answers. There are 3 of these quizzes because there are 3 different websites. And like I said, you may use them. It's certainly optional. Any question about any of this material? For each lecture, there is a lecture worksheet. On the worksheet, this one I think you got to download. Nope, the questions are here. What you should do to answer the question, take the question in order. It looks like some people have already started. So the next question would be question, a lot of people have started, question 9. What you should do is copy question 9. You only need to do one. And then put that in the start of your answer, which, where do we go for putting in an answer? Sorry, I'm not used to doing this. Reply, I guess.
That's right. Click on reply.
And then paste in the question, which I thought I had, and then type your answer, and then submit, post reply. And we're not going to do this because this is a question somebody can answer, but we will actually just post a period. And then take a look at it. It'll give it at the very bottom. I'll delete this. And make sure your question hasn't been answered, because if your question has been answered, you will not get at least full credit. You may not get any credit. I think I have the ability to delete that, so I'm going to delete that. There we go. And then make sure your answer is correct. Look it up if you need to, because you have to give a correct answer the first time in the lecture. If you don't like the question, like you don't know what bright field illumination is and you don't want to look it up, you can just let another student answer this, come back to the quiz another time, and someplace down the line, you'll get another question, and you might get with a light microscope. Most microbes are colorless and have little contrast to the slide. It's hard to see the cells unless they're stained. Why does staining the slide help? And you say, I know this one. And then you copy it and then put in the answer and submit or post the answer. You only need to do one question, and all of these are numbered because there's about 20 students in the class and there are only 20 questions. Any questions? All right, I think that's it. Let's go ahead and continue with the lesson. I will mention we are having a lab today, and that will be lab 4 on stains. So we had begun talking about simple lipids. They're called fats or triglycerides, and they are made up of glycerol and then fatty acids attached to the glycerol. Triglycerides are the most common fat in the human body, as well as in our diet. Another lipid is a phospholipid, and it is an unusual lipid in that it is not totally nonpolar, meaning a phospholipid is a polar molecule, and it is also nonpolar, which makes this molecule biphasic. Biphasic molecules are not terribly common, but I'm sure you're familiar with the biphasic molecule, and that's because soaps and detergents are biphasic. And that's why detergents wash our clothes better, because detergent is biphasic. One part of the detergent can dissolve oil in our clothes, and then the other part of the detergent can dissolve in water, meaning it's polar, so it dissolves in water. And that's why washing our clothes with detergents cleans the clothes better than just washing the clothes with water alone. With the detergent in there, we can remove the water-soluble molecules as well as the lipid-soluble molecules. Any question about that part? If not, let's return to the phospholipid. It is a lipid, but it has a phosphate group on it, and the phospholipid has a polar part, the head of the phospholipid, which contains the phosphate group. Let me see if I can blow this up. And not only is this molecule polar, it's actually ionic, meaning the phosphate molecule has a charge. But the phosphate group is polar, making the head of the phospholipid polar. And the phospholipid, we're not showing all of the molecule. It has the lipid soluble portion, which is actually two fatty acids linked to a glycerol. The glycerol is in the head, and the glycerol is partly polar. And attached in the third position of the glycerol is the phosphate group. And then attached to the phosphate group is an R group. The R group is some organic molecule, and it can be essentially anything, and we're just abbreviating it as R. Any questions about any of that? Anyways, in a phospholipid, this region of the molecule is polar, the head, and then the tails, the fatty acid portion, is nonpolar. The tails can dissolve in lipids. The head can dissolve in water, making phospholipid a biphasic molecule. When we put phospholipids in water, the molecules like this, come on mouse, will start lining up like that, where the tails will be on the inside and the heads will be on the outside, because the heads like being around water. We're putting the molecules in water. And then the tails are away from the water on the inside. We show you the next molecule. And the phospholipids will actually start forming a bilayer like this, which will then go around, forming a liposome, where we have water inside the phospholipid layers, so we call it a bilayer, and then water on the outside. What does this remind you of? Anybody? What shape is it? Somebody gave an answer. I didn't hear it. All right. It should remind you of a cell. And the phospholipids are the main molecule in the membrane of eukaryote and bacteria cells. It in archaea is a molecule similar to phospholipid, but it is not phospholipid. And we'll briefly talk about that in some future point. The point is that this should remind you of a cell, and the phospholipid is the main molecule in the membrane of a cell. And inside the cell, we have the cytoplasm, which is mostly water. And then outside the cell, if we're talking about something like blood, or doesn't really matter because outside the cell, the body is mostly water. Any question about any of that? State that it always amazes me when I think about our hand, that we're animals, so our cells are held together only by the cell membrane, which is mostly phospholipids. And this molecule is actually at the temperature of our body, is actually a liquid. And so this liquid is holding my hand together. And that always amazes me. Any questions about any of that? All right. If not, let's move on to the steroids and the sterols. They are a normal lipid in the sense that they are nonpolar. Steroids and sterols have 4 interconnected groups, which are ribbon carbon strings, carbon ring structures. If the steroid has an OH group attached to it, we then call that a sterol. And sterols are an important part of the animal plasma membrane. So there would be an animal plasma membrane, and there you can see the phospholipid bilayer making the membrane. But it does have sterols, particularly cholesterol, inside the membrane. And the important part of cholesterol in the membrane is it gives strength to the membrane. Here's a question for you. Cholesterol is found only in what foods? What cells? What tissues? Where do we find cholesterol?
In fat?
You do find it in most fat, but not all fat. Like you would not find it in palm oil or coconut oil.
Is it unsaturated fats?
No. You can find cholesterol in unsaturated fats and saturated fats. Like when we have beef fat, most of it is saturated. But when you have pork fat, some of that fat is unsaturated. And you can find cholesterol in pork fat. So I told you you can find it in beef fat and pork fat. So that's giving you a hint. Where do we find cholesterol? Where do we not find cholesterol? Well, for those of you who are vegetarians, or if you eat a vegetarian meal, you're not going to get cholesterol because cholesterol is only found in animal cells. And yes, cholesterol is usually found in fats, but only in animal fat. So like butter or lard, for example, will have cholesterol. And then coconut oil, which is actually a solid, would not have cholesterol. Something different about cholesterol, let me shrink that. And that is steroids and sterols are not made of monomers. The simple fats and the triglycerides are made up of monomers, as well as phospholipids. They're made of monomers too. But steroids and sterols have no monomers. There's one cholesterol molecule, and that's all there is, at least for cholesterol, which I think there are different types of cholesterol, but you're getting the point. All right, any questions about lipids? If not, let's turn on to proteins. Proteins are essential in cell structure and function. Proteins may have diverse chemical properties, giving them diverse functions. For example, there are structural proteins found in bone, muscle, and hair. There are enzymes, which mostly are proteins, which help speed up chemical reactions. There are transport proteins, which move a specific chemical from one side of the membrane to another. For example, we have a protein in our cell to move glucose inside the cell, and that would be a transport protein. There are regulatory proteins which regulate the body function. Examples would be different hormones, or at least some hormones. There are proteins responsible for movement. The reason why we walk or the reason why bacteria swims is because of proteins. Proteins are responsible for some bacterial toxins. We'll talk about toxins later in the term. For now, all you need to know that toxins are bad things and they're poisonous to us. And some of these toxins are various proteins. And then lastly, there are antibodies, which are proteins that our bodies make to help defend us against foreign invaders. Any question about proteins? You may have noticed that proteins are a little more diverse than other of the biological molecules. The reason why they are more diverse is because they are made up of monomers, and these monomers are called amino acids. And generally for the lipids and the carbohydrates, we have a handful, at most a dozen, of the different monomers which make up the carbohydrate or the lipids. But for proteins, we have 20 different amino acids that can make up the protein. And so proteins are more diverse because they have more diverse monomers. A monomer or an amino acid has the general shape shown here, and that is we have a carboxyl group in the center. On one end, we have, well, let's go ahead and talk about the H. On one end, we have a hydrogen bound to it. On the other end, we have an R group. An R group can vary depending on which amino acid we're talking about. Now, there are 20 different amino acids that humans have, so there would be 20 different R groups in humans. Any question about that part? On one side of that central carbon of the amino acid, we have an amino group, which is either NH2 or NH3 with a positive charge. And on the other end, away from the amino group, is our carboxyl group, which can be CO with a double link, OH or COOH, or it could be COO with a negative charge. Usually when that hydrogen comes off, the electrons stay with the electron, with the oxygen, sorry, the electron stays with the oxygen, making COO have a negative charge. And then that hydrogen just comes over to the amino group, changing it from NH2 to NH3 plus. Amino acids are linked together to form a protein, and you just hook one amino acid to another to get the protein. Any question about any of that? Oh, wrong one. This slide is showing you some of the different amino acids. They only differ in their R group. You don't need to memorize any of these. The simplest R group is glycine, which simply has a hydrogen for the R. See if I can blow that up. So it looks like that's the R group here because it's circled. And then we have an R group with CH3, and then an R group which is CH with two CH3s combined. All of these molecules, and this one here too, are these R groups are nonpolar. However, the R group doesn't have to be nonpolar, like in this one, tyrosine. This is the R group, highlighted in darker blue. This region of this R group is polar, the OH group. And because of that, we say that the R group is polar, although a large portion of this R group is nonpolar, meaning this region here and that region there. Here's another polar R group. You don't need to know the names. This is an histidine. There are 10 other R groups that are not shown for a total of 20 different amino acids. Any question about the R groups or the amino acids? Amino acids are linked together one after another to make the protein. When we start linking the amino acids together, like glycine and alanine, they are linked together by dehydration synthesis. So water comes out, OH from one amino acid, hydrogen from the other amino acid, and then that N directly links to that C, that CO group, OH group, sorry, COOH group, right there. And that link we call a peptide bond, because this link is actually fairly rare in the human body as well as in the universe outside of peptides. You might remember a peptide is a short chain of amino acids. It might be in it might be a protein that's being formed, in which case it's not fully formed, in which case we call it a peptide. Any question about any of that? So proteins are a polypeptide chain, and they are composed of monomers that we call amino acids. Blow this up. There are 4 levels of protein structure: the primary level, the secondary level, the tertiary level, and the quaternary level. Let's watch a little video on that, if it plays. And the primary level is just the sequence of amino acids after the protein is first made. Looks like we have a commercial here.
As I'm in the midst of applying for residency right now, wish me luck, Grammarly has again been a lifesaver with my application.
Hopefully I can.
In this video, we're going to talk about the structure of proteins. And what basically is a protein? A protein is a polymer consistent of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein. Whenever you have many amino acids, it's also called a polypeptide. And the bond that connects each individual amino acid residue is a peptide bond. So let's talk about the individual structure of an amino acid. So an amino acid has a chiral carbon with a hydrogen attached to it, and it has an amine group attached to it as well. And then it has an R group and a carboxyl group. Now I'm going to draw another amino acid right next to it. The left side, where the nitrogen is located, is the N-terminal of the amino acid. The right side, that has the carboxyl group, is the C-terminal of the amino acid. Now these two amino acids can react with each other in a condensation reaction where we're going to lose water, and we're going to connect the two amino acids into one molecule. So once water is removed, we could form a peptide bond. This is also called a dehydration reaction because you're losing water. And so this is going to be the new product of the reaction. So let's see if I can fit it in here. So this oxygen is gone. So now we have a nitrogen. Let me take out H2O. We have one hydrogen left over. We lost two once we lost water. So this is a dipeptide because we have two amino acids combined. And so this amide bond that we see here, that is the peptide bond. We formed a bond between the carbon and the nitrogen atom. So whenever you combine two amino acids to form a peptide bond, you're forming a covalent bond, which is hard to break. Now let's talk about the different levels of protein structure. There's 4 levels that you need to know: the primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure. Now the primary structure is simply based on the sequence of the amino acids found in the protein. The sequence determines the shape of the protein. If you replace just one amino acid with another, it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein. And there's 2 of them that you need to be familiar with: the alpha helix and the beta pleated sheet. In the alpha helix, you could see that it's stabilized by hydrogen bonds. The NH group of one amino acid interacts with the carbonyl group of another amino acid. And each turn contains about 3.6 amino acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure, which represents the 3-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we could see some areas is just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet. Now, the tertiary structure is one individual subunit. When you combine multiple subunits, you create a quaternary structure. So hemoglobin is an example of that. It has 4 individual subunits, 2 alpha subunits and 2 beta subunits. So hemoglobin is a protein with a quaternary structure. And so that's basically it for this video. Hopefully it gave you a basic understanding of proteins and the 4 levels of structure that they have. Thanks for watching.
All right, any questions?
I had some questions about the secondary structure. Can you clarify a little bit more about it? He went a little fast in the video.
Okay. All right, well, let me just start with the primary structure and then I'll move on to the secondary structure. The primary structure is just the sequence of amino acids as the protein is made. The secondary structure happens because the nearby amino acids in the protein start interacting with each other. There can be local interactions, meaning nearby amino acids interact with each other. And that can give rise to 3 different shapes. I think the video may have only talked about 2. There's the alpha helix shown here, the beta pleated sheet shown there. And then the 3rd is just a random coil, meaning the protein can take on any shape. This happens because of local interactions, meaning nearby amino acids are interacting. It can happen by hydrogen bonds forming between 2 amino acids shown here. The red one is hydrogen bonding to the blue one and vice versa. It can happen because of nonpolar, polar interactions, meaning a repulsion. If this molecule is polar, sorry, my mouse has died. If this amino acid, crud, if this amino acid is polar and that one is nonpolar, these two will be repulsed from each other. If this one is polar and that one is polar, they'll be attracted to each other. But then again, actually, it'd be that if they were polar and polar. If they're nonpolar and nonpolar, they will have an attraction for each other. And how you can illustrate that is you can put like a little blob of butter in oil or mix two oils together, and they will interact with each other. They will be attracted to each other. And we're not going to talk much about that nonpolar attraction, but it's called van der Waals forces. So they're attracted to each other. There's also ionic attractions. If this amino acid is, now let's go with this one. If this one has a positive charge and that one has a negative charge, then these two amino acids will be attracted to each other. And that's an ionic attraction. There can also be ionic repulsion. If this one has a positive charge and that one has a positive charge, these amino acids will be repulsed from each other. All of these interactions can happen. And let me warn you that I think your textbook and the video itself mainly talk about the hydrogen bonds being the only interaction that's happening. That is not correct. There are many interactions that can happen at the secondary structure of a protein. In fact, all of the interactions happen except for one interaction, which is seen in the tertiary structure. But it is important to remember that the secondary structure is happening because nearby amino acids are interacting with each other on the protein chain. So nearby amino acids. Now, the tertiary level is where amino acids from all over the protein can interact with each other. For example, this protein is being attracted, this amino acid is being attracted to this amino acid while they're coming together here. And this one is near the end of the protein, and this one is near the beginning of the protein. So the tertiary structure has interactions between all the different amino acids all over the protein chain, and the secondary only has nearby interactions. The tertiary structure also gives the overall three-dimensional structure of the polypeptide chain. So the polypeptide chain was initially one amino acid laid out after another, and then the chain can bend and fold and interact with each other like this, taking on the three-dimensional structure of the protein. And you can call that the protein has actually supercoiled and is now interacting all over it with each other. One interaction that you can find in the tertiary structure and you do not find in the secondary structure is disulfide bridges. The amino acid cysteine has a sulfhydryl group on it. Let me end this and I'll do it in Word. So this will be the protein here, and this will be a protein here, and let's just go down like that. And we have the amino acid cysteine, and it has a sulfhydryl group, S and H connected, and what happens is we have another chain over here. I don't know if you can see where my hand was going, but so we're going to put a sulfhydryl group again. And then the cysteine. CYS stands for cysteine. The hydrogens can come off going into solution, and then the two sulfurs can link together. And I don't know how to do that. I'm just going to erase that. And that is a sulfhydryl group where this chain here is linked together with this chain here by a covalent bond forming between these two sulfur groups. This is a really strong interaction. How you can learn that it's a very strong interaction was my mother used to put her hair in a curling iron and then put, what do you call it, bobby pins and whatever those things were called, bobby pins and something to hold the hair on the roller. So it's a roller and a bobby pin. And then she'd put on a cap and then steam her hair. And then she would take it off, and that would be like 10, 15 minutes later, and then take out the roller and the bobby pin, and her hair would have a wave. And that was a hydrogen bond. And then she would go into the Western Washington or Western Oregon weather where there'd be mist in the air and that curl would sag because the water, just the mist in the air, would be enough to interact with that hydrogen bond that she made to form the curl. And so the curl would sag. On the other hand, if you go in and get your hair permed, it will directly create the cysteine sulfur bridge where the two sulfurs come together and causing the disulfide bridges. Whoop, I'm on the wrong slide here. Why did I get out of there? Which is the disulfide bond, and it's very strong. You can go into the shower for a month and the perm of your hair will be fine. Any questions about that? So the hydrogen bonds are very weak. They break and reform all the time. The disulfide bridges are very strong. That's a covalent bond. And that is what's holding the different amino acids together in the protein, a covalent bond. The hydrogen bonds are very weak. But with the tertiary structure, you can find all of the interactions you found in the secondary structure, meaning you can find hydrogen bonds, you can have hydrophobic interactions, you can find ionic bonds and ionic repulsions. You can have not hydrophobic, a polar amino acid being attracted to another polar amino acid, and then a polar amino acid being repulsed from a non-polar amino acid. Any question about any of that? All right. So many proteins, the highest level you find to the protein structure is the tertiary structure. Like most enzymes, as high as they can go is the tertiary structure. But some proteins have the quaternary structure, and these are proteins that are made up of at least 2 or more polypeptides. And then the polypeptides come together and then interact with each other from all of the interactions we discussed with the tertiary interactions. And then they come together and form the final protein. Like hemoglobin and antibodies are made up of 4 polypeptides that come together to form the finished protein. And so hemoglobin and antibodies have a quaternary structure. Any question about that? Catalase and catecholase, which you may be studied in Biology 160, and most enzymes for that matter, only go up to our tertiary structure because they only have one polypeptide making the entire protein. All right, so you need to know that proteins can take different shapes. We can have a globular protein, and these are compact, roughly spherical proteins like hemoglobin. And then we can have fibrous proteins. They're more thread-like, and fibrous proteins typically are in, what do you call it? Fibers or structural proteins like collagen. We can have other molecules conjugated to proteins, and we call these conjugated proteins, where the amino acid portion, the protein portion, links with a covalent bond to an organic or inorganic compound. These are named by their non-protein component, like a glycoprotein is a protein plus a glycogen, which is a carbohydrate. So glycoproteins are protein and a carbohydrate bound together. Lipoproteins, which I'm sure you've heard of, are protein and a lipid linked together. And you can find lipoproteins and glycoproteins in the blood as well as in the cell membrane. When we're talking about proteins, you should realize that the three-dimensional shape is important for the functioning of the protein. And if the protein loses its three-dimensional shape, either breaking hydrogen bonds, like when you heat up a protein, or maybe by forming hydrogen bonds, like when you freeze a protein, when the hydrogen bond shouldn't be there, you can have them form. That will cause loss of the protein's three-dimensional shape, loss of the protein's function. The protein, if it loses its shape totally, we'll say it is no longer functional, and we call it denatured. Proteins can become denatured whenever they're in the wrong environment. So proteins have to have the correct temperature, the correct pH, and the correct salt concentration. And if you put them in an environment at the wrong temperature, the wrong pH, or the incorrect salt concentration, the protein will start unwinding, unfolding, and that we call denaturing, where they lose their three-dimensional shape. All right, any question about proteins? If not, let's move on to the fourth of the biological molecule, the nucleic acids. There are two nucleic acids you need to know, DNA, or deoxyribonucleic acid, and RNA, which is an abbreviation for ribonucleic acid. DNA and RNA consist of monomers that we call nucleotides. So nucleic acids are the polymer, and they're made up of monomers called nucleotides. You should know that a nucleotide itself is made up of three different parts. There's a pentose sugar, that's a 5-carbon sugar. There is a phosphate group, it has a charge to it. And then there's a nitrogen-containing base, shown, or one of the bases is shown right here. When we're talking about a nucleic acid in a single strand, it is held together by the direct linking, meaning a covalent bond between the sugar and the phosphate groups. The base is held off on the side, And then the base can link together with another DNA strand, if this is a DNA strand, and they will link together, but only linking together by hydrogen bonds. So the bases link together, and there are specific bases that link together, meaning we call them, certain bases are complementary to each other. The A and the T always link together, or the T and the A. And then the C and the G always link together, or the G and the C. When two nucleotide strands are held together, they're held together by hydrogen bonds, which form between the bases. All right, in DNA, there are 4 nitrogen-containing bases, or 4 nucleotides. And in RNA, there are also 4 nucleotides, but you do need to know that RNA substitutes 1 nucleotide so that it's not the same 4 as in DNA. The 4 nucleotides in DNA are adenine, or A, guanine, or G, cytosine, or C, and thymine, or T. And I don't care if you know them by adenine or the single letter abbreviation A. You do need to know one of them. So you should know that DNA has 4 nucleotides, A, G, C, and T. Okay. And you should also know that A and T complement, meaning they hydrogen bond with each other, and they form only 2 hydrogen bonds between A and T, or T and A. And then C and G are complementary, and they form 3 hydrogen bonds between each other. Let me show you the A and T bond here. So this is adenine here, and this nitrogen hydrogen bonds to the O and thymine, the T here. And then this nitrogen and adenine hydrogen bonds to the NH group on the thymine here. Any questions about hydrogen bonding, complementary bases, and all that? All right, let me take a step back and say you do need to know that A and G are purines in DNA, and C and T are pyrimidines. And an easy way to remember this is pyrimidine has a Y in the name. Cytosine and thymine have Y's in them. So cytosine and thymine are pyrimidines. Adenine and guanine do not have Y's in them, and they are not pyrimidines. They are purines. Okay. Now for RNA, they have the 3 of the 4 the same nucleotides. They have the A, they have the G, they have the C, or cytosine. They do not have, RNA does not have thymine. Thymine is replaced by uracil in RNA. Okay. And then the complementary basing pairing in RNA is that A and T don't pair because T isn't in RNA. It's A and U that pair. And how you can remember that U is a pyrimidine is U replaces T, and thymine is a pyrimidine, and both pyrimidines and thymine have a Y in them. Okay? Any question about any of that? All right. So let's look at this strand of DNA. You should know that DNA is a double strand, 2 strands, and it's held together by the sugar, which is deoxyribose for DNA, and then the phosphate. So here we have a sugar, there we have a phosphate, there we have a sugar. Actually, that's a, well, sugar phosphate hold the single strand of DNA together by a covalent chemical bond. The two strands of DNA are held together only by hydrogen bonds between the nucleotides, the nitrogen-containing bases. So C and the G form 3 hydrogen bonds, A and the T form 2 hydrogen bonds, and they hold the two strands together. So one strand of DNA is held together by covalent bonds, but two strands are held together only by hydrogen bonds. So the hydrogen bonds are much weaker than the single strand of DNA. And you can make DNA single-stranded just by heating it up, something like 70 degrees Celsius, I think, will make DNA single-stranded, because the heat will break the hydrogen bonds, but not the covalent chemical bonds in the single strand. DNA, you should know, is twisted in a ladder shown here, and it is the bases which make the rungs in the ladder. Any question about any of that? All right, RNA, shown here, has the sugar ribose, which all you need to know, the ribose has an extra oxygen in the 5-carbon sugar than deoxyribose. And RNA is single-stranded, so only one strand of RNA exists. It's not double-stranded. RNA, the single strand, is held together by the sugar phosphate backbone. You should know that there are 3 major kinds of RNA: tRNA or transfer RNA, mRNA or messenger RNA, and rRNA or ribosomal RNA. All 3 kinds of the RNA are important in protein translation. Here's a question for you. How do the tRNA, messenger RNA, and rRNA differ? Do they have different nucleotides? Do they have different sugars? Do they have different bases, which would be different nucleotides? The answer is no. They all have the same sugar, ribose. They're all single-stranded. They all have the same bases, the 4 bases. Let's see if I can remember this right. G, C, A. They don't have T, they have U that replaces T. So the only difference in the 3 different major types of RNA is the sequence of nucleotides. And the sequence of nucleotides determines if it's a tRNA, a messenger RNA, or an rRNA. All right. We need to talk a little bit about ATP. ATP is adenosine triphosphate. ATP is a molecule of the nucleotides, not the nucleotides, the nucleic acids, meaning it's in this family, but it's simply a triphosphate nucleotide, meaning there are 3 phosphate groups attached to it. ATP is the primary energy source for cells, meaning whenever a cell needs energy, it typically gets its energy needs from ATP. ATP does have adenine as the nitrogen-containing base. It does have ribose as the sugar, and then it has 3 phosphates attached. This phosphate group and this phosphate group have are bound by a high energy bond. So that's a high energy bond, that's a high energy bond, and that's abbreviated by a squiggly line. Whenever the cell needs energy, it will break this bond, releasing a lot of energy, and then the cell can use some of that energy for the cell's energy needs. If the cell needs further energy, then this one bond can provide, the cell could break that bond to generate more energy. ATP is made by dehydration synthesis. What we do is we get ADP, or adenosine diphosphate. You add on an inorganic phosphate group, meaning a third phosphate, and then you add on energy, and that will form water, and then this phosphate will bind to the ADP to make ATP. And then you can reverse this by hydrolysis, adding on water, and then the reaction goes this way to make ADP. You break off the phosphate group, the third, and then that releases energy. And like I said, whenever the cell needs energy, it'll break this ATP group to release this energy. All right, we talked about hydrogen bonds in this lesson. What molecules that we discussed have hydrogen bonds that give the molecule important properties? Anybody?
Water.
Water is definitely a molecule that has important properties because of hydrogen bonds. How about DNA? Could you have a double strand of DNA if you didn't have hydrogen bonds? Nobody's going to guess. Okay. All right. You guys can think about that question. A lot of the molecules that we discussed today had hydrogen bonds, including water, which we discussed last time. Carbohydrates have hydrogen bonds. Proteins have hydrogen bonds. Phospholipids have hydrogen bonds, but that's about the only one. And then nucleic acid have hydrogen bonds. And what would happen to these molecules if the hydrogen bonds were to suddenly all disappear?
Would they all become denatured?
Well, proteins would become denatured. The other molecules wouldn't be interacting correctly. Water, for example, if all the hydrogen's liquid water, if all the hydrogen bonds were to break, the liquid water would become gaseous water. Okay. But yeah, they're important for the functioning of these molecules. And so our molecules would not function, or they would, like water, it would totally evaporate from our body, and we would not have life if that were to happen. All right, any questions about chapter 2? If not, let's go on to chapter 3. Chapter 3 is observing microorganisms. Know the terms of this chapter. Understand the sizes of organisms and the properties of microscopes. Understand the major differences between a light microscope and an electron microscope. Understand why staining improves resolution under a microscope. And know that there are 3 types of stains: a simple stain, a differential stain, and a special stain. And then lastly, understand the Gram stain, know its procedure, and the basis of its differential staining. Any question about what we're going to be doing? All right. You need to know the metric system because we use it when we're observing microorganisms. You need to know it for a few other reasons too, but that's why we're discussing it now. You should know the units of measurement. We, in science and medicine, use the metric system. For example, the meter is the base unit of the metric system to measure length. And you should know that 1,000 millimeters equal 1 meter. You should know that 10 to the 6, well, excuse me, I think I said that backwards. No, that's right, 1,000 millimeters, 1 meter. 1,000 micrometers equal 1 millimeter. You don't need to know that one, but you should know that 1 micrometer equals 10 to the negative 6 power meters, meaning you have 1,000,000 or 10 to the 6 micrometers in 1 meter. Okay. And then with the nanometer, you should know that you have 10 to the 9th nanometers equal 1 meter. Any question about that? You are expected to know this on quizzes and exams. We do have the metric system quizzes to help you learn that. They're optional quizzes. And this table you need to know. Let me see if I can blow this out. Move this up here. So you should know that when we're talking about length, we have a kilometer. Kilo means 1,000, and there's 1,000 meters in 1 kilometer. And then we talked about meter. You don't really need to know a decimeter, but you should know a centimeter. There's 1 meter is, what's an easy way to word this? It isn't worded that way. So let me just go this way and say that we have 1/100th or 0.01 meters equals 1 centimeter. A millimeter is 1/1000th of a meter, or 0.001 meters equals 1 millimeter. A micrometer is 10 to the negative 6 meters, meaning 0.00000, if I said that right, 1 meters equals 1 micrometer. A nanometer is 10 to the negative 9th meters, and you don't really need to know a picometer. Okay, so switching between metric, once you learn it, is actually easy. You just need to know the suffix. Kilo is 1,000, centimeters 1/100th, millimeters 1/1000th, micrometers 1/1,000,000th or 1 to 10 to the 6th, and nanometer is 1 billionth. Is that right? Or 1 to the 10 to the negative 9th. Yeah, that's a billion. That's a million. So switching between the metric, once you learn the prefixes, is easy. And then when we're talking about weight or mass, we don't use meters, we use grams. And then for volume, we typically don't use meters, we use, what is that called? Sorry, I'm losing it. We use liters. There we go. But you use the same prefix. So there would be a kiloliter, a centiliter, a milliliter. And they're all the same. Metric units is set up so that 1 gram of water is equal to 1 milliliter of water. And 1 milliliter of water is equal to 1 cubic centimeter of water. So 1 cc is cubic centimeter. It equals 1 milliliter. Any question about any of that? All right, you are expected to know that on the quiz. And this slide is showing you why you need to know the metric system, because this map is showing you all the countries in the world that have not used the metric system as their primary means of measuring things. And as you can notice, there's only 3 countries in the world that do not use the metric system. And all of them are small, except for the United States. And I can say that the United States is slowly converting to using the metric system. It was actually Thomas Jefferson who signed the legislation, I think he proposed it too, to have the United States use the metric system. And as you know, that was a long time ago. The United States is very, very slowly switching over to using the metric system. But you'll notice on the cars and most signs, they will tell you the miles per hour, or the sign will tell you, well, either the miles per hour or how many miles away it is, but it'll tell you also how many kilometers per hour you're going, or how many kilometers away something is. So we're slowly starting to sign over to using the metric system. And that's one reason. And the other reason why you need to know the metric system is because science and medicine use the metric system. Like when you're going to give a shot to someone, you're going to give it to them in millimeters or cc's, which is cubic centimeters. You're not going to give it to them in inches, for example, or in, what would it call, the teaspoon? Give the child a teaspoon injection. They don't do that. You should know the typical sizes of microorganisms, and basically you just need to know that eukaryotic cells are on average about 10 times larger than a prokaryotic cell, because everybody has a different size range, 1 to 10 micrometers, a prokaryotic cell, and actually they can go less than 1 micrometer. A eukaryotic cell, it depends on what cell you're looking at, but average 10 micrometers to 500 micrometers. And if you don't know, the largest cell that you've ever seen is the yolk of a chicken egg. And it's much larger than 500 micrometers. The yolk of an egg is practically an inch, and that's about 2.4 centimeters. Any question about that? We're not really going to be talking much about viruses. Later in the term, we'll talk about viruses, but for now, you just need to know that viruses are about 10 times smaller than the smallest prokaryotic cell. It depends on what virus you're talking about. The very largest virus, the length, but not the diameter, well, actually the diameter too, can approach the size of a small prokaryotic cell. You should know the different sizes of things. We have things which are visible, like a person, an egg, a frog egg, about the smallest thing you can see with your naked eye. And then we use the microscope to see most eukaryotic cells. As far as I know, all prokaryotic cells. To see smaller things, like viruses, proteins, large molecules, small molecules like water, and then atoms, you need a light microscope. Not a light microscope, an electron microscope. Do you remember that there's a mistake in this table? It has 1 micrometer here and 1 micrometer there. This one is correct. That should be a millimeter right here. So when we're talking about microscopes, you should know that in the lab, we have compound microscopes. They have 2 or more lenses that magnify. The total magnification being the magnification of the objective lens you're using times the ocular lens. In our lab and in most microscopes, they simply have a 10x ocular lens. And so the total magnification will be 10 times whatever objective lens you're using. In our lab, for the objective lens, we have 4x times magnification, 10x times 40x times, and 100x times. So the total magnification would range from 40x times to 1000x times. Any question about any of that? If there's no questions, I'm going to stop here and I'll see you at 6:30 for the lab. All right, bye.